Okay, this is our last installment for the muscle tissue lecture. And so we had just finished talking about the sarcomere and the sliding filament model of contraction that explained to us how the sarcomere shortens to ultimately create this muscle contraction. So we were looking at the microscopic level in the last lecture. For this part, we're going up to the gross anatomy level, and we're going to think about the muscle as an organ, really, and how it creates the kinds of contractions and the amount of force that is necessary for muscles to do what they do. And of course, that's going to be different for the different muscles of our body because they are responsible for different kinds of movement, like moving an eyeball versus picking up something really heavy, like a barbell. And we're going to be examining some of this stuff in lab as well with our skeletal muscle physiology lab. Okay, so we want to identify a few terms here, define a few terms. So a motor unit refers to one motor neuron and all of the muscle fibers that it innervates. So when we first started and we looked at that neuromuscular junction, we saw a picture of a motor neuron coming down and splitting up into a whole bunch of axon terminals. And each of those axon terminals was able to supply one muscle fiber. So a motor unit is that one motor neuron and all the fibers that it innervates through those many, many axon terminals. And the picture on this screen is showing you that same thing as a drawing and also as a photomicrograph, so something kind of under the microscope. And that picture on the right might look like those familiar skeletal muscle cells that we've looked at under the microscope. And then you can see a whole bunch of those axon terminals coming off of that motor neuron to supply a whole bunch of them. So one nerve could have hundreds of motor neurons in it. When we talk about the nervous system, we'll explain that a nerve is made up of a whole bunch of neurons, axons. So one nerve can have hundreds of motor neurons, and then each neuron can supply many, many muscle fibers, and that would be the motor unit. And so the number of fibers per motor unit varies quite a bit. Could be 4, could be all the way up to 400. And again, that's going to depend on the kinds of responsibilities that the muscle has, right? And this is really an example of structure reflecting function. So fine motor control muscles, muscles that are just going to make small and subtle, precise kinds of movements, such as moving the eyes, as I mentioned before, or maybe some of those muscles in your fingers that carry out the precision grip and enable you to write in cursive. Those kinds of muscles are going to have small motor units, that is, fewer fibers per neuron. And that's going to give that fine motor control to the muscle. And larger muscles that carry out kind of greater strength kinds of activities that are less precise, like your quadriceps and your thighs, for example, are going to have larger motor units, so more fibers per neuron. So we want to be aware of that potential distinction when we're thinking about an individual muscle of the body. Okay, in your electromyography, EMG, skeletal muscle physiology lab, we're going to be looking at a muscle twitch. And a muscle twitch is really a single response from one stimulation. And this really never happens in your body because there's always many, many neurons firing and many sources of stimulation to an individual muscle. And so it's kind of a lab phenomenon that we use to understand how innervation reaches muscles and how muscle strength and contraction results from that. When we look at a muscle twitch, we tend to measure the muscle tension or the force exerted by a contracting muscle on an object. And the object is basically the load. So if you're thinking about the lever system, which describes how a machine or describes the mechanical way that movement can occur across a joint, for example, or a fulcrum, as it's called in the mechanical example. The object that we're describing here is known as the load. That's the opposing force exerted on a muscle. So if you are trying to lift a pile of dirt with a shovel, the pile of dirt on the big end of your shovel would be the load. So the muscle tension is the amount of force needed by a contracting muscle to move that object or to move that load. So we're going to measure this up the y-axis when we show the data of a muscle twitch in a visual representation, which is called a myogram. And so there's our root for muscle, myo. And a myogram is what you're seeing in the picture in the bottom right. That's the recording of the contractile activity of a muscle. And the line that really makes the myogram is known as a tracing. And that's a term that you may come across when you are doing your electrophysiology lab this week or next week. So a myogram basically has 3 phases. So there are 3 parts to a myogram that we can identify. What's called the latent period, and this is the first few milliseconds because typically time is going to be measured, the duration of the muscle twitch is measured along the x-axis, as you can see on the myogram, and it's typically measured in milliseconds. So these are pretty quick occurrences. So the first few milliseconds that follow the stimulation is known as the latent period. So if you look at the myogram, the stimulus is the stimulation, and that's where the red arrow is showing you, and that's happening at our time 0. And then it's a very, very, very short, almost horizontal line on our tracing, and that very small part is the latent period. So the stimulus has occurred, but there isn't enough force for muscle contraction to be observable on our myogram. So excitation, contraction, coupling is occurring. So remember what that is. That's when the neuron came down, and after the event of the neuromuscular junction, that end plate potential occurred and caused the sodium channels to open and let sodium into the muscle cell, and that generated the action potential, which would then trigger the rest of the events that resulted in the muscle contraction. The action potential travels along the sarcolemma, down the T-tubule, releases calcium, etc. So as that is beginning to happen, that's the latent period. And when it generates enough force to be visible on our myogram, then we have our contraction period beginning. So during the period of contraction, enough cross bridges are active that tension can be viewed. And that period of contraction, you can see that curve rises up and reaches a maximum tension. And at that maximum tension, that's the greatest force that that muscle contraction can produce. So during your period of contraction, that's when you've got all that cross bridge cycling that we looked at in the last lecture. All of that is occurring. The period of relaxation, notice it begins right at the peak where our max tension has occurred during contraction, and then it continues all the way until that line decreases and reaches back down to 0 tension. And so the period of relaxation is occurring as calcium is being pumped back into the sarcoplasmic reticulum. Remember, that's where the calcium is stored. And so contractile force is declining because remember what calcium does. Calcium binds to the troponin, causing the tropomyosin to shift, revealing the myosin binding sites on the actin. So without calcium, those myosin binding sites are not available, and fewer and fewer and fewer cross bridges are going to be able to form. And it's the cross bridges that create the sliding of the filaments and therefore the contraction. So muscle tension will eventually decrease all the way down to 0, back to the baseline during the relaxation period. And you can see that contraction occurs much faster than relaxation. The relaxation period is the longest period in the whole duration of the muscle twitch. So you'll be looking at a lot of myograms in lab. So again, that's what happens with one individual stimulus causing a muscle contraction. And we use it as kind of our baseline to understand the actual phenomenon that occur in the body. And so a graded muscle contraction is really what's happening in your body when your muscles contract. This is describing a normal muscle contraction that is going to vary in strength based on need. That's what graded refers to. The variation is why we call it a graded muscle contraction. So again, the amount of force you need to pick up a coffee cup is not the same as the amount of force you would need to pick up a 20-pound barbell. And so the muscles have to adjust accordingly so that you have a kind of smooth and natural movement, not kind of a jerky response movement. And so these graded muscle contractions are going to really be what your body demonstrates. And so that grading, that kind of variation that occurs as necessary depending on the muscle and the situation and the load, right, occurs in two ways. So graded muscle contraction occurs through what we call temporal summation and through what we call recruitment. And so temporal always refers to time. So temporal summation is telling you that there's an increase in the frequency of stimulation. There's an increase in the number of times that a stimulus occurs within that muscle contraction. And so the rate of firing of action potentials along a motor neuron is going to increase. Okay? So temporal summation, increase in the number of times that our stimulus occurs. Temporal summation is also called wave summation, and we'll see why when we look at the picture. And then the other way that muscle contraction is graded is through recruitment, or sometimes we see that as motor unit recruitment. And so recruitment is going to increase the strength of the stimulation, which will increase the number of motor units activated. And remember, a motor unit is one motor neuron and all of the muscle fibers that it supplies. So the picture again is just showing you another muscle twitch. And again, time is measured across the x-axis. You should know that's the horizontal axis, and tension is recorded along the y-axis. That's the vertical axis. Time is measured in milliseconds. And so we see the latent period and the stimulus, and then the contraction rising to maximal tension, which they're showing you with that white dotted line. That's as high as that peak is going to rise. That's the maximal tension that can be produced from that stimulus. And then you have your relaxation period, and then it just kind of plateaus at 0. Okay, so let's think about these a little bit more. And I should mention that temporal summation and recruitment are just controlled by your brain. It's not really something that you really willfully control, though we do have voluntary control of our skeletal muscle. These mechanisms are just occurring kind of involuntarily to create whatever strength contraction is necessary. So temporal summation is also called wave summation, because when you look at the picture of the myogram of this phenomenon, it very much looks like waves. And so we can start to follow a normal kind of myogram. So we have our latent period, and there's the stimulus and the latent period, and then the contraction period. We see our peak for that first tracing. And then as we begin to relax, notice we never get back down to that 0 because another stimulus is introduced. And so that's going to cause another contraction period. And notice that this time we achieve a higher tension than we did the first time. And then we start to relax, but then another stimulus is introduced, and that creates an even higher peak for that part of the contraction, and so on, until there's no more stimulus introduced, and then we have a normal relaxation back down to the base. So if a second stimulus occurs before relaxation is complete, then the second wave is stronger than the first, and that's why it's called wave summation. We're kind of summing or adding up the strength of the individual contractions, and that's how we get an even stronger contraction, which we might need if we are lifting something heavy. And so the reason that we're able to achieve a higher tension, greater strength with each additional stimulus is because the muscle is already partially contracted. So we already have a bunch of those cross bridges formed, and so that's already a certain level of strength. And then we introduce even more of those cross bridges to make a stronger contraction. And then the other consideration is that, remember what happens during relaxation, the calcium is going to be pumped back into the sarcoplasmic reticulum for storage. But if that process is interrupted by another stimulus, then there's already still a bunch of calcium available to bind to the troponin, to cause the tropomyosin to shift, to expose the myosin binding sites, and therefore allow more cross-bridge formation, which is going to be a stronger contraction. So that's what interrupting the relaxation period does. That's why it allows for the next wave to be higher, to be stronger, have greater tension. So this process increases the force of the muscle contraction, allows for greater strength, but also results in kind of a smooth, continuous muscle contraction. So like we said, we want it to be kind of fluid, right? You don't have jerky movements as you try to lift something. You can do it pretty evenly, pretty easily. And that's because of this process that is allowing kind of a gradual increase in strength as necessary. So you get, a nice normal contraction as we think of it, as we view it. And the degree of wave summation will continue until something which is called incomplete tetanus is achieved. So incomplete tetanus, you'll look at this in lab, also called unfused tetanus, is basically where you have the stimulation coming so quickly that the relaxation is very, very, very short. And you end up with kind of a quivering, sustained contraction, as it's often described. So we'll look at that, I believe, in lab. Unfused tetanus can lead to fused tetanus. So when you look at this myogram, right, we have time in milliseconds on the x-axis and tension on the y-axis. We have our 0 time, and then every red arrow indicates a stimulus. So here the stimuli are coming very, very, very rapidly. In fact, they're coming so fast that eventually there's no time for any relaxation to occur at all. And so this results in a sustained muscle contraction. This really doesn't happen very often in the body because we have other kinds of mechanisms that prevent this from happening. So this isn't really a very effective process for our muscle contraction. And the word tetanus might be familiar to you because of getting a tetanus shot, but that is something very different. That tetanus is a bacterial disease that can cause various symptoms such as intense muscle spasms. And of course, if those muscles spazzing are your muscles of respiration, like your diaphragm, that can prevent proper breathing and can, in some cases, be fatal. So we don't want to confuse that tetanus with this sort of muscular phenomenon that really we see in the lab, but normally don't see in a human body. Okay, so remember, we're talking about getting those graded muscle contractions where we're able to vary the strength of the muscle contraction as needed by the body. And the first way that we did that was through temporal summation, varying the frequency of the stimulus to build that wave-like contraction with greater and greater strength, higher force in each subsequent wave. The other way that we do that we create these graded muscle contractions, is through recruitment, sometimes called multiple motor unit summation. So you see that term motor unit again, and that's why we wanted to be clear on what that means. A motor unit is one motor neuron and all the muscle fibers that it innervates. So here we're looking at 3 different pictures that are representing the same phenomenon. So at the bottom, you have the myogram as you've looked at before. So time across the x-axis, tension on the y-axis, okay? And you can follow that tracing in blue. And we'll talk about that more in a minute. And then the middle picture is showing you the cross-section of a muscle or of a muscle with the various motor units excited. So you can see the cross-section of the muscle on the far left has no dark circles. That means no motor units are excited or active. And then all the way on the right, you see that they're all active. And between none and all, there are varying degrees, right? So we're going to talk about that. And then the top picture is also following through time. That's shown on the x-axis of the myogram. And it's showing you the strength of stimulus. And so in lab, when we demonstrate stimulus, it's through voltage. Remember, that's kind of your electricity. So here we're looking at the frequency of stimuli to the nerve. So every arrow again indicates a new stimulus. And then this time on the y-axis, we have the voltage. So really the strength of that stimulus. And so we can look at these three pictures kind of together. So let's identify some other terms. So a subthreshold stimulus is one that is not strong enough to produce an observable contraction. So if you look at the top picture, number one, that's the first stimulus. Look at the middle picture, you've got no motor units activated. And if you look at the myogram, it's still a flat line at 0. So that was a subthreshold stimulus. Remember, sub means below. Okay, so a threshold stimulus is the one we need that has enough strength to show a visible peak or visible contraction on the myogram. Okay, so subthreshold is below that amount that we need. So you can see again in the top picture, 1 is subthreshold. If you look at the cross-section of motor units, none are excited. 2, stimulus 2 is subthreshold. Again, no motor units are excited. And on our myogram, we have a flat horizontal line. But then stimulus 3, which is greater than 1 and 2, begins to show us a little bit of that muscle twitch on the myogram. So we do see a visible peak, and I've put it in a green circle on that bottom myogram. And in the middle picture, we see 2 of the many motor units have been recruited. That means they've sort of been called into action. So the stimulus at 3 is our threshold stimulus. It was the amount of voltage required to cause a visible contraction, to get some of those motor units to become active. And then 4, you can see, is above our threshold, as are 5 and 6 and 7. And so we have higher and higher and higher peaks as the stimulus is becoming greater. And you can see in that middle picture, more and more and more motor units are recruited, are called into activity by that stimulus. When we reach stimulus 7, that is our maximal stimulus or maximum stimulus. That is the greatest strength of contraction that we can get. So notice on your myogram on the bottom picture, that's the highest peak that is generated. And it's the same height peak that stimulus 8 generates, that stimulus 9 generates, that stimulus 10 generates. So it didn't matter how much stronger the voltage was, how much stronger the stimulus was. After that maximal stimulus, we couldn't recruit any more motor units for any greater strength, right? So the last 4 cross sections in that middle picture, all of the motor units are colored in, they've all been recruited. If all the motor units are recruited, that's the greatest strength that we're going to achieve. So maximal stimulus is the strongest stimulus that increases contractile force. So even though we had stronger stimuli, numbers 8, 9, and 10, it didn't result in a stronger contraction because all the motor units were already active. Okay, so stimulating beyond that point does not give us a stronger contraction. So threshold is what we need to show a visible contraction to get some motor units active. Subthreshold means anything below that stimulus voltage, nothing's going to happen. Maximal stimulus is the strongest stimulus that will yield contractile force. Beyond that, nothing new is going to happen. So we're going to look at these phenomena in our physiology lab online, and it's pretty user-friendly. It really walks you through. It tells you exactly what to click, what to look at, what to do. It's kind of a step-by-step simulation. Okay, so shouldn't have too much problem with that as long as you have watched this lecture and kind of gone through these notes and read this part of the textbook and are familiar with these kinds of terms. Okay, so motor unit summation or recruitment. The more motor units we call into activity, the stronger the muscle contraction. And there's what's known as motor recruitment size principle. So it's not random which motor units are called into activity first. The first group that become active are the smallest fibers, the smallest muscle fibers. So remember, a fiber is a cell. So the smallest muscle cells are highly excitable, and they're going to become activated first in the recruitment process. Second, the medium-sized ones are next, and that will really add to the strength of the contraction. And then lastly, the largest motor units are going to be engaged, and only when a really powerful contraction is necessary, like if you were using your thighs to lift one end of a couch. So that's the size principle: smallest first, medium second, and largest last. And typically, not all motor units contract at the same time. Some are resting while others are contracting. And that enables us to maintain strong, prolonged contraction and kind of allow our bodies and our muscles to do all the things that we constantly require them to do. Okay. And it also allows an increase in weak contractions to be gradual and an increase in strong contractions to be progressively greater when larger amounts of force are needed. So again, we're able to deliver the correct amount of strength for whatever activity we're doing, right? So we use the same hand and the same muscles in the arm to gently touch somebody's cheek as we do to punch somebody in the face. But clearly the strength of muscle contraction is not going to be the same. We have much more control and a much gentler touch when we're touching somebody's face lovingly than we do when we use those same bones and fingers and joints to punch them. And that's because of this recruitment principle. We start with the smaller ones, and we really only use those big, 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 strong ones when we absolutely need them for that particular kind of activity. All right, when we talk about these kinds of contractions, we can distinguish between what are called isometric and isotonic contractions. And we have some zoological roots here helping us remember what these words are all about. Iso means same, and tonic or tonicity, and we'll talk more about that in the next lecture, refers to tension. Remember, we talked about muscle tension a little bit, muscle tone, as that slight contraction that occurs because some motor units during relaxation are still going to be active. And this helps to stabilize our skeleton, helps to keep our joints aligned, and also keeps the muscle ready to kind of carry out a larger contraction when necessary. So they're kind of ready to respond. So in an isotonic contraction, muscle tension is able to overcome the load resulting in the muscle shortening or lengthening. So look at the picture on the left, and this is what you're going to see when you do your simulation in lab. So you have a muscle there, and at one end of it is a 3 kilogram weight. And really one end of a muscle is attached at the origin and the other end is attached at the insertion. I've thought about this before with muscles. And we know that the insertion end is typically the end that moves. So the insertion end in this case would be where that weight is handled. And so the muscle contraction has to be strong enough to move that weight, to overcome the load, which in this case is that 3-kilogram weight. So when muscle tension developed is able to overcome the load, it's going to pull the insertion end towards the origin end, moving that weight, and the muscle shortens. Okay, and that shortening is what we described the sarcomere doing at the microscopic level. Now we're looking at a big muscle shortening at the gross anatomy level. And the best demonstration of this is when you're in anatomical position and you flex your forearm, your nice biceps brachii bulges out, and that visible bulge is there because the muscle itself shortened due to all that sarcomere stuff that we talked about in the last lecture. So the thin filaments slid towards each other, as we described, moving the Z discs together, shortening the sarcomere, the muscle contracts enough to move that load. Okay? So this is what happens when you're lifting your textbook, for example. Notice that the slide says, when muscle tension developed overcomes the load resulting in muscle shortening or lengthening. So there's an ideal length to a sarcomere for contraction, and sometimes it shortens, sometimes it actually lengthens. Okay? It stretches beyond that ideal size. And so when this happens, the lengthening is called an eccentric contraction, ECC, ENTRIC. What they're showing you here is a concentric contraction. Those are the shortening contractions. So lifting your A&P book is a concentric contraction because your muscle is shortening. You could see again your biceps brachii bulge. But walking down a hill would be an eccentric, where you have that kind of extra lengthening. And eccentric contractions are kind of like braking muscles. You know, when you're walking down a hill, your quadriceps are kind of making sure that you don't tumble forward with the gravity and the slope of the hill. Okay. But let's take a look at the myogram here. So now we have two graphs to look at. On the top is the myogram. So you've got time going across the x-axis in both of these pictures. And then on the y-axis up top is the tension. And so we have a somewhat normal myogram there. We have our latent period, our stimulus, our contraction, and then the peak tension is maintained, and then you see the relaxation. Okay. The bottom graph is showing you the length of the muscle. Okay, so it starts at 100%. That means that it's at its full length. And when the muscle contraction is at peak tension, notice the length of the muscle decreases. It goes from 100% length down to about 77% length. So that's your muscle shortening during peak contraction. Okay, so you kind of want to look at those two together to understand how the muscle shape is changing. So that's a standard isotonic kind of contraction. Isometric, so those words are telling us same measure, or really same size. So in an isometric contraction, the muscle tension that develops is not strong enough to actually move the load, so the muscle stays the same length. So when you look at the picture on the left, there's that muscle again, origin at the top, insertion where the weight is. This time it's 6 kilograms. So when the stimulus comes down and causes this muscle to contract, it can't overcome the 6 kilogram load, and it does not shorten. It doesn't move the weight, and the muscle doesn't shorten. So it stays the same size, isometric. Okay. You're going to still have cross-bridge formation here, but the thin filaments aren't able to be pulled towards each other, so there's no sliding and there's no shortening of the muscle. Think about if you push a wall. If you put your palms against a wall and you push, you're definitely contracting muscles. You can feel the muscle tension, you can feel the muscles contracting, but you're not moving anything. Okay, so those muscles aren't going to shorten. This is how your postural muscles operate because they are just creating enough muscle tension to help keep things in place. They don't actually want to move things, move parts of your body. When you're maintaining posture, you're sitting still, you're maintaining a position. Okay? And these muscles that act as fixators are doing the same thing. So if they're holding a muscle in place, they are holding a bone in place, I should say, they don't want that bone to move. So the contraction of the muscle is going to be an isometric kind of contraction, kind of like a clenching sort of force. Okay. And so again, if we look at the picture on the right, we have a somewhat typical myogram on the top. So we have our latent period, our stimulus, we have our contraction rising, and we have our peak resistance, and then we have the relaxation back down to zero tension. On the bottom, during the duration of that muscle twitch, you can see that the length of the muscle starts at 100 and never changes. So it doesn't get any shorter. Remember in the isotonic contraction on the previous slide, that green line dipped down and the muscle length got shorter. Okay, so for example, if we were standing under a bar, standing under a chin-up bar, we would have a lot of eccentric contraction, kind of holding our joints together, or I'm sorry, isometric contractions, holding our joints together, maintaining our posture. If we then grab the bar and take our feet off the ground, so now we're hanging from the bar, these are isometric contractions because you know your muscles are contracting, right? You have to hold yourself there, but your muscles aren't shortening because you're not overloading the weight of your body. The tension is not greater than the weight of your body that is hanging there. If you then do a chin up, now you're demonstrating isotonic contractions. Okay, you are going to have shortening of muscles, bulging of muscles as you lift the weight of your body. So you do overcome the load with that chin up. And then as you lower your body back down, still hanging from the bar, those are going to be eccentric isotonic contractions. So now you're lengthening the sarcomere, so changing the shape so it's still isotonic, but in a lengthening kind of way. Okay, so that's one example of kind of isolating the three sort of variations of contractions: isometric, and then concentric isotonic, and eccentric isotonic. Okay, so we have a little check-in quiz here. You know what to do at this point. You've got 5 questions. So take a minute to answer them. Maybe pause the PowerPoint presentation because I'm going to go ahead and go on to the answer slide in 3, 2, 1. Okay, so one motor neuron in all of the muscle fibers that it innervates is called a motor unit. Right? A muscle twitch is just that single muscle contraction from one stimulus. The myogram is the picture of that muscle twitch, and the load is just the force that is opposing the muscle twitch. Number 2, which period lasts the longest in a muscle twitch? Relaxation was definitely the longest. The latent period was the shortest, and contraction typically in between. If a muscle shortens when it contracts, this is a isotonic contraction. So remember, same tension, isotonic, not same size, which would be isometric because it shortened. The first fibers to be recruited in a muscle are the smallest. It goes smallest, medium-sized, and then biggest. It is not random in your motor unit recruitment. And then finally, temporal summation results from a increased frequency of stimulation. Remember, temporal means time. So we're talking about more times that our stimulus is delivered. Okay. Now, we know that ATP is required for muscle contraction. ATP stands for adenosine triphosphate, and it is the cellular energy that we know mitochondria produce, organelles inside of our cells, and muscle cells do tend to have a lot of mitochondria for ATP production. And we talked about ATP being necessary in cross-bridge cycling in the way that the myosin attaches to the actin that requires an ATP, and then the release of actin from myosin requires an ATP. We may also have mentioned that ATP is necessary to get that calcium out of the cytosol back into the sarcoplasmic reticulum for storage in the terminal cistern during the relaxation period of a muscle contraction. And then also, and we'll learn more about this, ATP is necessary for the sodium-potassium pump, which is going to be important in moving sodium ions and potassium ions to opposite sides of the plasma membrane. Okay, so we know we need ATP, but where does it come from? Very little ATP is actually stored in your cells. Once it is broken down into ADP, adenosine diphosphate, and that single phosphate, it's almost instantly rebuilt. So our cells have very good mechanisms for producing more ATP when the ingredients are present to do that. So we have two categories of ATP production: anaerobic and aerobic. And aero, A-E-R-O, means air, in this case referring to oxygen. So aerobic mechanisms require oxygen. Anaerobic, A-N, one of your negative zoological prefixes, meaning without oxygen. So anaerobic means do not require oxygen, and aerobic means do require oxygen. Okay, so simply, in simple terms, one means of anaerobic respiration, or anaerobic ATP production, I should say, is described as the direct phosphorylation of ADP by creatine phosphate. So that's a mouthful, but basically creatine phosphate is a high energy molecule that we have in our muscles. And in the presence of ADP, adenosine diphosphate, when there is an enzyme called creatine kinase, and notice that ASE, which often tells us that we're talking about an enzyme, the creatine phosphate is broken down into creatine and a single phosphate. And that single phosphate, of course, is going to combine with the ADP to give us ATP. So creatine phosphate and ADP will yield creatine and ATP, because ADP, adenosine diphosphate, plus one more phosphate gives us adenosine triphosphate. And this does not require the presence of oxygen. You don't see that anywhere in the equation. So it is an anaerobic process. And this direct phosphorylation provides maximum muscle power, but only for about 15 seconds. So it's not an extremely long duration of energy. And you get 1 ATP for every creatine phosphate molecule. So that's not a really great yield. We're going to see that our cellular respiration gives us way more than that. It gives us 32 ATP for every glucose molecule. So this is not our most efficient. It's a low yield. We don't get very much ATP out of it, and it's only good for short spurts, but we can do it in the absence of oxygen. It's a reversible action, so we can rebuild our creatine phosphate for future breakdown. And this reversal tends to happen during rest or inactivity, so when your muscles are not actively contracting. Okay, our second form of anaerobic respiration, so without oxygen, is glycolysis. So we see glyco, right, glucose in there. and lysis, breaking down. So we're breaking down glucose, glycolysis. And so that's the first phase of glucose breakdown, because a couple of different things can happen here. It's anaerobic, does not require oxygen. And so glucose is broken down into two pyruvic acid molecules, which releases enough energy to make a small amount of ATP. So in this process, we get 2 ATP per glucose. So that's a little bit better than the 1 ATP we got from our creatine phosphate. And then pyruvic acid is further converted into lactic acid. And lactic acid is moved into the bloodstream and picked up by some other organs of the body, which can use it as an energy source. For example, the liver can reconvert lactic acid to pyruvic acid or into glucose. So we store a lot of glucose in the liver through this mechanism. For example, during rigorous exercise, muscle contraction constricts blood vessels, so less oxygen reaches muscles. So when you're contracting muscles in an isotonic contraction, they may be shortening, and that shortening can create a bulge to the fibers in the muscle, and that can constrict blood vessels. So if you think about that biceps brachii, when you flex your forearm, you get that huge bulging biceps brachii. Blood vessels can be temporarily constricted when that happens. No blood vessels, no blood supply, less oxygen reaching the muscle. So that's when these anaerobic mechanisms are going to be important. So this anaerobic glycolysis is fast, but again, not very efficient because it only gives us 2 ATP per glucose. And as I mentioned, we can get 32 ATP per glucose in our aerobic cellular respiration. So it's a low yield, but it's a good process to be able to use in the absence of oxygen. Excuse me. Okay, but most of the ATP for our muscles comes from aerobic respiration. So we know that we do need oxygen here. So when oxygen is present, the glycolysis occurs, but pyruvic acid is moved into mitochondria instead, and this produces even more ATP through aerobic respiration. So we can start with that, excuse me, same kind of glycolysis, but with oxygen, the pyruvic acid can be moved into the mitochondria and cellular respiration can occur there. So glucose, now in the presence of oxygen, will yield carbon dioxide, water, and ATP. And notice we get 32 ATP per glucose with this reaction. So that is way more ATP per glucose molecule than we had in our anaerobic forms. And we see those waste products, carbon dioxide and water. We've talked about how those are relatively easy to get rid of. Carbon dioxide through our respiratory system, exhaling, and water through sweating, urinating, defecating, and exhalation as well. So this kind of respiration, aerobic, provides the most energy. However, it has a whole bunch of steps. So guys, this is your Krebs cycle. If you've ever had to take a cell biology class and go through all these steps, there's a lot happening. So it's a slow process. It requires a lot of oxygen and a lot of glucose. So we're going to see that muscle fibers that are carrying out this cellular respiration are going to have a lot of mitochondria, but also a lot of that myoglobin for oxygen, and maybe even a lot of those glycosomes for glucose storage. So during exercise, we can use glycogen that was stored in the muscle. That's going to get used up first. Remember, glycogen is the stored form of glucose. And then glucose does arrive to our cells through the blood supply. So that would be the likely second source. And then pyruvic acid from glycolysis and free fatty acids can further be used for energy production for the muscle cell. So aerobic respiration, cellular respiration, makes the most ATP for our muscles. So this is just kind of showing you again the order that we might, our muscle cells might use these different kinds of ATP production through the course of exercise. ATP in the very beginning comes from that stored glucose. And then we're gonna typically engage the creatine phosphate breakdown. And then a little bit later, for the duration, the glycogen stored in muscles is going to be broken down when oxygen is present to generate ATP. For prolonged duration exercise like marathon running, ATP is generated by the breakdown of several nutrient fuels by aerobic pathways. So some of those other acids or carbohydrates or sometimes proteins are going to be used for duration exercises. Okay, so we know that muscle fatigue is a physiological phenomenon. So we're not talking about you feeling tired. We're talking about actual physiological muscle fatigue where the muscle isn't able to contract even when it is stimulated at threshold levels. And this phenomenon is not completely understood or well understood, but muscle fatigue tends to be prevented in the body from reducing total ATP stores because without any ATP, muscle cells can die. So some of the things that seem to influence muscle fatigue include ionic imbalances. So sodium, potassium levels are really important for our muscle contraction and our neural conduction, as well as calcium. So if those things are off, that can be a problem. And increased inorganic phosphate. So if too much phosphate is present, it can interfere with kind of the regulation of calcium. And we know why calcium is so important for our muscle contraction because of its role in exposing the myosin binding sites on actin. Decreased ATP and increased magnesium. So magnesium seems to have an effect on the voltage sensitive proteins that regulate calcium release. So that can be a problem, again, not enough calcium. And decreased glycogen, of course, because we need a source of glucose for ATP production. So there's lots of different factors, and it's not completely understood exactly how it all works. Muscle fatigue is not the same as when your muscles get sore after a workout. Muscle soreness is believed to be caused more by micro tears in the muscle cells. So that's more of a physical as opposed to physiological deficit. Okay, so we understand that we need different forces to our muscle contractions depending on what we're going to do. And so that force of contraction is due to the number of cross bridges attached. Okay, so the more cross bridges attached we have, the more sliding filaments are going to move and the stronger the contraction will be. So there are 4 factors that affect this, and we've already talked about 3 of them. So frequency of stimulation, right? The more frequent stimuli we have, the greater the force because of the formation of that temporal summation that we talked about. Motor unit recruitment, so the number of muscle fibers stimulated, which is called recruitment. The more motor units that are recruited, the greater the force. So the more fibers that are engaged, right, the stronger the contraction will be. And then we mentioned the size of fibers in that recruitment. So the bulkier the muscle, the more tension it can develop. So muscle cells can increase in size. That's called hypertrophy with regular exercise. So when we build muscles, we're not making more muscle cells, but we're developing more myofibrils inside the muscle cell. That's hypertrophy, right? Trophy refers to kind of nourishment. So hyper, above regular nourishment, we're going to have bigger muscles like this Arnold Schwarzenegger here. And of course, that hypertrophy can be reversed with disuse. That would be atrophy of muscles. So the fourth factor that we haven't discussed yet is the degree of muscle stretch. So we talked about the sarcomere at rest having a particular length, and then we talked about during contraction how the sarcomere length shortens. And so this degree of muscle stretch, where the muscle fibers have sarcomeres at different lengths, influences the strength of the contraction. So if the sarcomere is less than 80% of that normal resting length, then the filaments overlap too much and force decreases. If the sarcomere is greater than that 120% of resting length, the filaments do not overlap enough, and so force decreases. Okay, so too short or too long decreases the efficiency, preventing, that cross bridge formation and the proper amount of muscle contraction force, or the optimal amount of muscle contraction force. So this graph is showing you on the x-axis the length of the sarcomere, and on the y-axis, again, we have that tension measurement. So at the optimal length, between 80 and 120% of the normal sarcomere length, we get our maximum tension, and then less when the sarcomere is too short and less when the sarcomere is too long. So you see those decreases from that optimal length. Okay, so those are the four factors increasing the force of skeletal muscle contraction. Large number of muscle fibers recruited, so motor unit recruitment, the large size of certain muscle fibers, high frequency of stimulation, so that was our temporal or wave summation, and then the muscle and sarcomere size in terms of its length, how much it has been stretched. And again, the greater the force is ultimately due to more cross bridges being formed. Okay, so when we talk about muscle fibers, skeletal muscle fibers, there are three categories, and they have different structural and functional characteristics. So we can talk about slow oxidative fibers, fast oxidative fibers, fast glycolytic fibers. So each of those categories has two words in it. The first is either fast or slow, and then the second is either oxidative or glycolytic. And so the fast or slow is describing the speed of contraction. And the second term, oxidative or glycolytic, is talking about the pathway of ATP formation. So that's how we get those names. And so you can see some of the differences here. Speed of contraction is slow, fast, fast, matching the title. And along with that is what's described as myosin ATPase activity, which is really just the speed that ATP is split to release energy. So we see ATPase, ACE being that kind of enzyme clue, meaning we're breaking something down. And then you can see the lineup of aerobic, and anaerobic, right? So if it's oxidative, you hear oxygen in there, so that's going to suggest aerobic to you because we need oxygen for aerobic mechanisms. Myoglobin content, remember what myoglobin does, it holds oxygen. So we're going to have more myoglobin in those oxidative processes than in the anaerobic process. That makes sense. And glycogen stores hold glucose. And so we're going to need that the most in fast glycolytic, where we are undergoing that glycolysis directly to produce our ATP. Recruitment order there, and then rate of fatigue. So slow oxidative has slow fatigue, so those are fatigue resistant, and that's because of that aerobic pathway. And then you can see intermediate and then fast. So the fast glycolytic fibers kind of burn out sort of quickly because the glycogen reserves are fairly short-lived. So just some things to consider about the different types of fibers that we find. The fast oxidative, those are the least common because they're sort of intermediate in everything. So this chart's kind of nice too. It highlights some of those characteristics that we see. And again, if we think about what they mean, it makes a bit of sense to us. The middle portion of this chart, activities best suited for. So that's something that you want to be able to recall. So slow oxidative fibers are going to be best for endurance type activities, running a marathon, maintaining posture, so your anti-gravity muscles, because they're fatigue resistant. They're going to be able to endure for long periods of time. Fast oxidative are good for kind of intermediate length and intermediate strength activities like sprinting and walking. And your fast glycolytic, where we get that quick but short burst of energy, is good for short-term intense or powerful movements like hitting a baseball or power lifting weights or something like that. Okay, so we know that we are supposed to be exercising, and that has lots of benefits to our body. In terms of muscle, we can do two kinds of exercises, sort of aerobic endurance exercise and resistance exercise. Endurance exercises like jogging and swimming and biking and walking lead to increased muscle capillaries. So we increase the blood supply to our muscles, which is good because that, of course, brings with it oxygen and brings with it nutrients and sugar, glucose, all of the things that those cells need. We can increase our mitochondria within muscle cells with these kinds of exercises, which of course allows us to produce more ATP for muscle contraction. And we can increase myoglobin synthesis and again be able to hold more oxygen in these muscles for more activity. So those require or result in greater endurance and strength and less fatigue in those muscle cells, in those muscles. And our bodies can actually convert the fast glycolytic fibers into the fast oxidative fibers. So the fast glycolytic fibers, remember, were good for the short, intense kinds of activities versus the fast oxidative fibers, which are better for more endurance. So we can convert one type of fibers to others with repeated exercise. Resistance exercise is really how we make our muscles bigger, like weightlifting or isometric exercises, where we're increasing the muscle contraction, but not enough to overload the force. So this will make our muscles hypertrophy, get bigger. And when our muscles get bigger, it's not because we're making more muscle cells or new muscle cells, but we're increasing fiber size by increasing the number of myofilaments within the muscle cells. We're going to get more mitochondria here too, more of those myofilaments, more glycogen stores, and more connective tissue to kind of help maintain the integrity of the muscle. Remember, muscles have 3 connective tissue layers: endomysium, perimysium, and epimysium. And this will result in increased muscle strength and size. If you've seen Arnold Schwarzenegger lately, he doesn't quite look like this. And that reminds us that muscle fibers can revert back to their original smaller size if these resistance exercises aren't kind of kept up with. And in severe cases, muscles can atrophy. So this is hypertrophy, but muscles can atrophy and become really deficient in size and function. And this tends to happen as we get older. So we always take a minute to talk about how things sort of decline as we get older, and that definitely applies to our muscle mass and strength. Our muscles get smaller and they get weaker, largely due to disuse or less use. And some of the active muscle tissue can be replaced with connective tissue or even fat. And of course, that's going to decrease their strength. But exercise is really the best thing that you can do to prevent this sort of decline in muscle structure and function. And remember, contracting muscles also helps to maintain your bones because that pull on the periosteum, for example, will stimulate osteoblasts to maintain bone tissue. Okay, so our last slide here is homeostasis. We want to think about how the muscle system works with other systems. And for this case, I've divided it up into muscle tissue types because we have 3 types of muscle tissue: cardiac, smooth, and skeletal. So cardiac muscle obviously is working with the cardiovascular system. As the myocardium, the middle layer of the heart wall, it creates a pumping action that's going to propel blood through the chambers of the heart, and then that creates the blood pressure that moves the blood through the arteries and through the capillary beds and through the veins of the body. Smooth muscle we're going to find in lots of other organs other than the heart. Hollow organs, for example, where it forms 2 layers in the wall to move the contents through. We call that peristalsis, but also has other food processing activities like churning and segmentation, also expulsion during defecation, and forms a lot of sphincters. And a sphincter is a ring-like smooth muscle valve that is going to regulate the passage of an organ's contents. Blood vessels have a smooth muscle layer, arteries and veins, and they create vasoconstriction and vasodilation. That is, they make the lumen smaller constriction and bigger dilation, and that greatly affects blood pressure and cardiac output, and also how much blood goes where. And there are sphincters in your capillary beds that also affect how much exchange occurs in a particular tissue. Ducts are going to have a smooth muscle layer. We have digestive ducts, reproductive ducts, urinary passageways, and they carry out peristalsis to move the contents through. And then our skeletal muscle, we know, moves the body, assists with breathing, locomotion, thermoregulates because heat is generated as a byproduct of skeletal muscle contraction, so that's going to warm us up. You'll see when you get to cardiovascular and lymphatic system that skeletal muscle acts as a pump to help propel blood against gravity back towards the heart, and same with lymph. And of course, it maintains our body's posture and helps to stabilize joints through muscle tone. Our skeletal muscle also assists with excretion, as we can use our diaphragm and our abdominal muscles to increase pressure in the abdominal cavity and help empty out those organs. Okay, last check-in for our muscle lecture. Here are your questions. Take a minute, pause the lecture, jot down your answers, and then go on to the answer page. Okay, processes that do not require oxygen for ATP production are called anaerobic. It's all in the zoological roots there, guys. Aero for air, in this case oxygen, AN meaning not, without. Which process creates the most ATP for muscle contraction? That is cellular respiration. That's what's happening inside your mitochondria, which is not a factor in force of muscle contraction. So muscle tone is the oddball on the list here. Degree of muscle stretch is important. Number of muscle fibers stimulated, that's your motor recruitment. Frequency of stimulation, that's your temporal summation, but not muscle tone. If a muscle fiber makes a lot of ATP through aerobic processes, it should have, now you have to think about this, aerobic processes mean we need oxygen. And so myoglobin A is going to store oxygen, so those kinds of muscle fibers need a lot of myoglobin. And then when your muscle gets larger, it is typically due to an increase in muscle fiber size. The other answers are all just nonsense. Okay, so that is the end of your muscle system lecture. The next lecture up just really organizes the various muscles of the body that you need to know in terms of origin, insertion, and action and location. And you'll be studying many of those muscles in lab with your lab work as well.